one way of thinking about social Hymenoptera is to think of them as having broken chromosomes. To recombine chromosomes in meiosis, you need to insert double-stranded breaks into them, or DSBs. Usually, the cell puts in more DSBs than are needed and chooses some to actually produce a crossover. In more recent data from Olaf Rippel's lab showed that Freesia mellifera also has a high recombination rate, not quite as high as the Apis group, but still quite high. So there is a general trend that's lasted over the 13 years since Wilford et al. Published, and it begs for explanation. Now, this is a quick overview of recombination and why it's hard to change. You need to have at least one crossover per chromosome, or the chromosomes don't separate properly, which is a common cause of infertility in women. In addition, crossovers are mutagenic. They cause because of errors in breakage repair. So as a result, in most organisms, there's crossover interference. The first crossover made tends to block remaining ones on that chromosome from happening. So all these constraints are there to keep crossover from becoming mutagenic, but somehow social bees escape the constraints. And I threw in a phylogeny there just because a lot of you are taxonomists and you'll go to sleep if I don't throw one in. How does evolution change recombination? Well, when Charlesworth and Hudson both published on what we now call background selection, they said, well, what if there are two mutants close to each other on the chromosome, linked? One of them's positive and one of them's negative. The negative one makes it hard for the positive one to be selected, and that's called negative background selection. So Sally Otto and Nick Barton worked hard on the theory side to see if there's a way to select for some kind of local modifier of recombination so that areas with such mutants can recombine more. And they found that it's very difficult, theoretically, to justify that. Meanwhile, over in the ant world, most of the buzz has been about inversions of the chromosome, which don't recombine with the other chromosome, and they therefore create supergenes. And so the picture below is, no, it's not a pollination network. It's a chromosome and a bunch of Formica ant species with a supergene area near the middle. So still no big recombination thing. So there are two hypotheses for the social bees. One is that there's relaxed selection and drift, effectively a loss of the constraints just due to small population sizes. And the second is that there might be positive selection for worker diversity. The relaxed selection and drift is because there's only one queen, even if there's thousands of workers in a colony. So ecological constraints mean that species with big colonies have fewer reproductives per unit area. And that makes specific predictions about what we should see in the population genetics of genes having to do with meiosis. If, on the other hand, more genetically diverse workers increase colony fitness, then either selection for increasing recombination in the queen or polyandry can increase colony fitness. Now, polyandry decreases relatedness between workers, whereas positive increasing recombination maintains worker relatedness. And so in many incipiently eusocial species, it's not a good idea to have polyandry because it blocks the worker relatedness, which helps create eusociality. So the predictions of the second hypothesis is that there should be signs of positive selection on genes for meiosis that increase double-stranded breaks and crossovers. Now, here's some unpublished data that I'm writing up that shows on two axes. Bottom axis is the protein evolutionary rate. So it's speaking to the question of, do these genes evolve more quickly? And the vertical axis is whether there's a correlation between a measure of selection and how social this bee species is. And this is done over 10 bee species. And in the upper right quadrant are the genes that the hypothesis 2 of positive selection would point to. And I've labeled a few of them. SPO11 is the gene that creates the double-stranded breaks, and the RAD51 and RAD9 genes are the genes responsible for helping turn them into crossovers. So they certainly fit the hypothesis 2 predictions. Now, the open question, though, is does worker diversity aid colony fitness? It's often asserted that it should, but there's not a ton of actual genetic and experimental data. So I've put up a picture from a paper that appeared this year in Behavioral Ecology, where using the same strains of Apis mellifera, some queens were mated 9 times and some were mated 54 times. And they measured a variety of things, including brood survival, which is on the vertical axis. And you can see that the queens that mated 54 times have significantly higher brood survival than the queens that mated 9 times. Now, there is this phenomenon of complementary sex determination, so queens need to mate at least a certain number of times, but that's usually thought to be 9 or 10 to reduce diploid males to a low number. So the increase in brood survival between 9 and 54 is not predicted by complementary sex determination. It must be due to something like increase in worker diversity helping with brood survival. So knowing that there is such a phenomenon of increased worker diversity helping to increase colony fitness, we can ask, is it true that increasing recombination will increase worker diversity? And in the interest of running through the slides quickly and answering questions, I didn't show you the disappointing cases that I modeled. My first model, you always start simply when you're modeling. I distributed genes for colony fitness in workers evenly along a chromosome and had polymorphisms in the queen in a number of locations. So she had different versions of those genes at a number of locations. And I did lots and lots of crossovers and measured what the worker fitness would be. And it did increase slightly when we went up to 1 crossover, but when we went up to 4 crossovers, there was no increase. And I was stumped for a while, and I said, but normally genes aren't spread evenly on a chromosome. And when I spread the genes unevenly on the chromosome and redid my simulations, you can see the 2 curves here. The top curve is with 1 crossover, and it shows the range of worker trait values in the colonies, the diversity of the colony in workers, with 1 crossover and with 4 crossovers. So yes, indeed, under certain conditions, if worker genes are not evenly spread out across the chromosome, then you can get increases in worker diversity by increasing the number of recombinations. So, where are we now? We've said increasing worker diversity can, in some cases, increase colony fitness. Increasing recombination can increase worker diversity. But now we go back to Otto and Nick Barton. They said it's really hard to select for modifiers of recombination on the chromosome. But recombination is a process. It's the intersection of multiple genetic pathways. The question I had was, could selection on 1 or a few genes change recombination rates? In other words, we're not selecting to increase recombination in one specific area. We're selecting to change recombination over the whole genome. Now, there's been indications in some of the studies done in honeybees that different queens have different recombination rates. And this paper published 2 years ago in Cell is the end product of a long investigation by this group. And what they found over quite a number of species, very distantly related, is that when you look at multiple different meiotic cells from an organism, crossover numbers will go up in one cell and down in the other cell, but they do so simultaneously over all the chromosomes. So this behaves as if there's a single pathway that governs the amount of crossover that happens. And it varies randomly between cells in the same individual. Now, if there's a single pathway, then selection can act on the genes in that pathway to select for more recombination in all the chromosomes of a cell or less recombination. And that gets rid of the problem that Otto and Barton first raised. So with that, I'd like to thank you for listening, and I'd like to acknowledge the funder, me, and answer your questions.
We have about 3 minutes for questions, and Clement is here to answer them. So let's see. Amro has a question.
Thanks, Clement. Fun talk. When you're doing the modeling, I'm just missing what's the mechanism where recombination increases working diversity? Is it because like recombination is mutagenic, or is just giving us like different haplotypes?
Yeah, it's I used a model. This will get a little bit techie for people who are not population geneticists, but I used a model of
Clement froze.
Oh no.
that has a small contribution to some worker diversity trait. And if the queen...
Sorry, Clement, you froze for like right as you were starting to talk about the model.
Okay, I'm going to stop my video and just try it with the sound. The model is one of many alleles of small effect. And the queen is polymorphic at, say, 100 sites on one chromosome. If we run crossovers on the queen's two chromosomes and look at the contribution of the meiotic product, so the workers' eggs that she laid, we can ask how much do their traits vary compared to the queen's parent chromosomes. So that's what I look at. And it's the range in that, because I'm trying to get at specifically the question of whether recombination increases the range of worker traits that I was graphing in those things. So not any mutagenic effects, simply reassortment of alleles into different worker chromosomes.
Got it. Thank you.